Today I will be talking about an article that examines if second language proficiency impacts networks in the brain related to language in bilingual translators. The article is titled Second Language Proficiency Modulates the Brain Language Control Network in Bilingual Translators, an event-related fMRI study. It was completed in 2019 by Mouthen and colleagues. Studying bilingualism and the brain areas associated with it has become an increasingly popular trend in recent years, specifically how the brain is able to organize multiple languages and use those languages accordingly. Previously, research on this topic has looked at a mechanism called language control to explain how bilingual individuals can communicate in one language rather than another. Many brain areas have been suggested to be a part of the network responsible for language control, such as the prefrontal cortex, anterior cingulate cortex, pre-supplementary motor area, left inferior parietal lobule, and left caudate nucleus. However, it is unknown whether there is a brain network solely used for language control, or if it is part of the highly connected language system in the brain as a whole. Also, it has been suggested through previous research that the control system is utilized differently depending on if someone is speaking their first or second language. Due to the lack of research dedicated to exploring the language control network in proficient versus non-proficient second language speakers, the aim of the study conducted by Mouthen and colleagues was to investigate whether language proficiency in bilingual speakers modulates to the language control system previously mentioned. The study was conducted with 30 female bilingual students at the School for Translation and Interpreting at Geneva University in Switzerland. They were all right-handed. This was because the brain behaves differently for left-handed individuals in terms of right versus left hemisphere activity. The students were split into two groups, with 15 students in each group. One group consisted of participants who were considered to have low proficiency in their second language, and the other had participants with high proficiency in their second language. Proficiency was determined by grouping those in their first year of their bachelor's degree at their school and those in their first year of their master's degree at the school. Then, two independent language experts evaluated the participants in order to ensure the students were meeting the expectations of language proficiency of the group they were assigned to. In the experiment, there was one experimental task and one control task. The experimental task involved the participants looking at black and white drawings of objects, an example being animals, clothes, or tools, while in an fMRI machine. In the simple version of the task, the participants would name the item in their first language. After about 4 minutes of naming the objects, participants would then have to name the object in either their first or second language, depending on the position of the drawing, whether it was in the upper or lower part of their screen. This was completed in about 8 minutes. The control task was similar, but instead involved letters and numbers. Participants first decided if the letter was a vowel or consonant, when shown a number and letter paired together. Then, based on the position of the stimuli, they needed to say whether the number was even or odd, or if the letter was a consonant or vowel. Participants' fMRI data were taken during both tasks, and also the reaction times and accuracy in identifying the stimuli. In order to look at the activation related to only language selection, the researchers took the brain imaging taken during the control task involving letters and numbers and subtracted it from the brain imaging taken during the experimental task involving naming objects in their first or second language in order to isolate what brain areas were related only to language selection. In the low proficiency group, there was brain activation in the middle frontal gyrus and left caudate that was not seen in the high proficiency group. There was also different left prefrontal network activation in those in the low versus high proficiency groups. On an individual level, it is also important to note that those in the higher proficiency group had higher accuracy in the task than those with low proficiency. The results of the study can inform the field of cognitive neuroscience of language about how the level of second language proficiency can impact how language control networks operate and can result in different brain activation during language selection tasks. The middle frontal gyrus and the left caudate activated for those with lower proficiency, but not with participants with higher proficiency in their second language. The researchers responsible for this study suggested that when someone has low proficiency in a second language, learning the second language requires more help from the brain than just general networks used to solve cognitive conflict. When someone becomes highly proficient in the second language they are learning, the cognitive control network in the brain is better able to handle challenges related to language selection because the individual is more experienced in their newly learned language. This conclusion can be summarized by stating that someone with low proficiency in a language needs more cognitive resources to manage language selection than those with a higher proficiency.